First of all, I would like to thank the organizers for giving me this fantastic opportunity to be being here. Even though I was not able to make it to the entire workshop, but I am still very happy to be able to interact with many of you now. Okay, so what I wanted to tell you about a little bit, well, this workshop, of course, is about prefrontal cortex. And one of the things that prefrontal cortex is known of, about many other things, is crucial role in working memory. So I want to try to break this concept a little bit, and we are thinking about how, especially is prefrontal cortex, I'm going to go in the other direction, how prefrontal cortex is not that special, because there's many other areas that also help in some of these functions. And I'm going to focus on working memory here. Let me just start with a basic idea that we all have. Working memory is just the cognitive function that allows us to retain and manipulate information during short periods of time. And since many years ago, and these are recordings from Joaquin Fuster, showing the correlation between this elevated or persistent activity in some neurons in prefrontal cortex and the maintenance of information in the system. And these neurons used to be recorded here in dorsolateral prefrontal cortex of the macaque in this case, and it was associated with the strong recurrent connections that we can find in pyramidal neurons in prefrontal cortex. This is the classical way that we have been seeing working memory for many years. However, we know that there's very recent evidence, or over the next 5 years especially, in which we see that this is not the entire picture. And working memory has been challenged in the last years in many ways. There have been models talking about silent working memory mechanisms, for example, or memory maintained in high dimensional spaces. But most of these new approaches focus on replacing the mechanism that we mentally place in dorsolateral prefrontal cortex. So I want to shake the tree in another direction, because it turns out that it's not only prefrontal cortex where we find signals or activity that is related to working memory. These are figures of a couple of recent studies. This is from, This is from a review from Christopher et al., where you see a collection of areas in the macaque cortex and the human cortex as well, in which activity related to working memory has been observed. And you see that it's rather distributed. There's nothing localized exclusively in prefrontal cortex. Last year, there was a really nice study by Voitov and Thomas Mersick-Flochel showing actually the evidence of the existence of distributed working memory in mice. So there's more and more evidence showing that working memory is not something that happens exclusively in prefrontal cortex. It also doesn't mean that happens everywhere, because this is, for example, a review by Julio Martinez Trujillo from a couple of years ago as well, 5 already, showing that there's a certain amount of evidence, positive and negative, for different areas in terms of the activity related to working memory. And we see that, of course, as we expected, the winners are prefrontal areas. Here they have a lot of positive evidence, let's say, for being involved in working memory, and also some temporal and parietal areas. And we have some other areas that are a little bit more controversial, like, for example, somatosensory cortex or primary visual. So this is tricky. This tells us that there's a certain level of heterogeneity at the level of the functions. And because of that, we start to look at the heterogeneity at the level of the anatomical properties of different areas. This is from a review from Martin van den Heuvel at the Vrije University of Amsterdam. And you can see that there is a lot of gradients regarding anatomical properties, structural properties that might be relevant for communication between neurons and maintenance of working memory in particular. For example, you can see that there's a nice gradient going from occipital to higher areas, frontal areas, in the size of the typical dendritic tree. that you see in pyramidal neurons, in the total spine count, spine density, and many other properties. Of course, this is kind of a challenge for the classical model of working memory, because, for example, if we focus on the number of spines that we observe in the basal dendrites in our pyramidal neurons, this is data that we collected from a collection of studies from Guy Elston. And you can see that, for example, there is, if you look at the, for example, areas like here, like early sensory visual cortex, V1, you can have up to 600-700 spines per neuron in the basal dendrites. But if you look at dorsolateral prefrontal cortex, this is almost 9,000. This is 1 order of magnitude above. So that means that if we want to use the classical attractor model for working memory, and we put our parameter for the coupling strength of these projections so that dorsolateral prefrontal cortex behaves well and is able to retain working memory, then all the other areas in the middle will not be able to effectively participate in working memory. And this goes against the evidence that we saw about many areas being involved in working memory, in simultaneous co-activation and these kind of things. On the other hand, we can also put some the constraints here, but then it will be an overactivation of higher areas. So there's clearly something that doesn't fit here. The classical local attractor model for working memory is not enough to explain the variability that we see in the new evidence. So this is where we started working on this. This was a project that I started while being a postdoc in the lab of Xiaojing Wang at New York University. And at this point, we were starting to think about how to explain this distributed activity related to working memory from a mechanistic point of view, using data constraint models. And I aligned data constraint because it was very important. On the one hand, we wanted to get the heterogeneity that we were finding in these studies about how some areas have more recurrent connectivity than others. And for this, we use the data for the spine count that we collected from Elston and colleagues. And on the other hand, we also wanted to have some data to constrain the interareal interactions between the different cortical areas. And for that, we use the data set from Henry Kennedy and colleagues. And fusing these two data sets, we were able to get, and of course, adding a local circuit that was able to show some level of competition between excitatory neurons coding for, let's say, a stimulus A, and another one's coding for stimulus B. And of course, with interneurons mediating this interaction. When we put all of this together, we have a large-scale model of the macaque brain that includes local area-specific heterogeneity and is able to tackle the question about how do these patterns of working memory emerge and whether we are able to explain them. And this is what the first thing that we did with the model. We just simulate a simplified version of a working memory task, a classical delay match to sample. And then you would have, for example, a queue that is here, for example, QA, or the green circle. The activity that we get in the model during the presentation of the queue is something like this. So you see a lot of activity in early visual cortex. However, if we look at the activity during the delay period, you see that there's a lot of widely distributed activation pattern that doesn't include areas like, for example, the primary visual areas. However, it involves a lot of areas, mostly on temporal and frontal lobes, and a little bit on parietal as well. This, of course, is a plain unselective pattern of working memory. Of course, if we want to simulate a working memory about remembering the color or an item, or a color of an item in the screen or something like that, then there would be particular areas that are more sensitive to encoding color cues, which would be more active, and other ones would be more inactive. But this is kind of an aggregate of what we would get in a widely generic working memory task. So it is fitting to compare it then with the evidence map that was collected with Julio Martinez Trujillo that I showed you a second ago. I'm going to put it here now in this nice format, and you can see that there is, of course, not a one-to-one correlation, but we get a good alignment with this evidence map from the experiments, from the experimental data, where we can see activation of areas in prefrontal and parietal cortex, especially frontal cortex, and very quickly, very few or none at all activation of occipital cortex, for example. Now, if you want to see a little bit more about the dynamics, this is how the traces about the firing rates of different areas look like. And you would see a fast activation in V1 mediated by the stimulus. And then when we take the stimulus out, we basically lose the activity. And we find similar things in areas like MT, but as soon as we go into higher areas in the cortical hierarchy, like for example, area LIP, we have a little bit of an integration with a peak here, dorsolateral prefrontal cortex, frontal eye fields, or anterior cingulate cortex. We see this level of co-activation and co-sustenance of activity related selectively to working memory. In this case, the green dot that we showed before. It's important to say that this was the extreme case, in the sense that we can always tune the model so that some areas, like say dorsolateral prefrontal cortex, are able to sustain this activity by themselves. But this is not the case. This is a case in which none of the areas that were in the model were able, when isolated, to sustain this selective activity. It's only because of the long-range interaction between the areas that the whole brain network is able to sustain this selective information in working memory. And as you can see, we get a very nice distribution here, some kind of a transition between areas that are able to sustain the activity and areas that are maintaining a spontaneous level. There's many things in this story that we can refer to, but I'm just going to go into some idea that I'm going to talk about a little bit later, which is the particularly important effect of feedback inhibition. There is something very important that we have to add to the model so that we are able to replicate the evidence map. And this is that we need to assume that there's some level of bias of the feedback projections. Those are the projections going from, for example, prefrontal areas to early sensory areas like V1. We need to assume that these long-range projections target inhibitory interneurons slightly stronger than excitatory neurons. This doesn't have to be a very big bias, but just with a little bit, we are able to see a lot of differences. For example, if we don't consider this bias at all, we will end up with a result of the simulations like this one, in which all of the areas are activated. And as you can imagine, this doesn't look anything at all like the experimental evidence. However, if we consider that, for example, consider that this is a higher area and this is a lower area in the hierarchy, if we consider that these projections from excitatory populations to the inhibitory population in the target area are a little bit stronger, then suddenly, if we don't have these projections, we have something like this. But as long as we start increasing the strength of this feedback inhibition, we go to a situation in which the areas at the top of the hierarchy, like prefrontal cortex, maintain a reasonable level of persistent activity, while early sensory areas that are at the bottom of the hierarchy, they go down to the spontaneous level that we know is present in the experiment. And just by looking at this, as we increase this level of feedback inhibition, we get closer and closer to the experimental evidence. And 100% is not a particularly big value, it's just the maximum value that I used, but it's not particularly large. Now, let me just get into the second part of the talk, because I want to tell you what did we find when we advance this idea and try to see whether it makes sense to think about distributed working memory in the human brain as well. And this is a job that I have done in collaboration with Menli Feng, a student in my lab, and Avirup Bandyopadhyay, a postdoc as well. This is at the University of Amsterdam, where I'm working right now. And the idea was to try to continue with this investigation and see if we can see patterns of distributed working memory in humans, because we know that in the experimental data we also find these kind of things. Of course, half of the story is to build the model itself, because as you may know, the data we can get for macaques and for humans is widely different. So we have to build the model entirely using different things. And in this case, we used 3 different data sets to constrain the model so that we are able to get a good level of predictive power, let's say. The first piece of evidence that we used was just diffusion-weighted imaging connectivity of the human brain, and this comes from the Human Connectome project. As you know, the human connectivity doesn't have information about directionality, and this is something very important to our model. So we wanted to add some arrow for the directionality. What is feed forward and what is feedback in the brain? And for that, we noticed that there has been recent evidence showing that, at least in macaques, the level of white matter in a particular area, as measured by tractography, correlates very well with the anatomical hierarchy. And this has been proposed in the Mejias et al. To have like a proxy, let's say, of an anatomical hierarchy in the human brain. And this anatomical hierarchy, this hierarchy based on white matter, or in particular by the ratio of T1 and T2, is the index that we are going to use to establish some kind of directionality in the circuit, in the network. And finally, we end up this collection of data with brain maps derived by PET scanners that show the relative density of NMDA receptors across different areas in the brain. And this is going to be very relevant because we know that NMDA receptors are going to be heavily involved in working memory. So when we collect all of these 3 data sets and we merge them together into a model, we are able to start investigating this aspect in the humans. And we are going to repeat the same idea. We just show a very simplified delay match to sample working memory task, very simplified version. And we get also that during the presentation, we get activation of early visual areas. And during the delay period, again, we have a distributed pattern of activity over many areas. Now, to compare it with experiments, we do something similar to what Martinez Trujillo and colleagues did in the review, collecting all the experimental evidence for the different areas in the macaque. But now we just did it for the humans. And we were able, after a thresholding of this data, we were able to compare this evidence map of the human with the results of the human model. And at this point, we were able to explain up to 72% of the variability of the data, that is the level of areas that are active or inactive in the human brain, with just tuning 2 global parameters, nothing else, that I'm going to show you right now. Of course, if you look at the traces, you get something very similar, elevation of activity in visual cortex, and areas like FEF and dorsolateral prefrontal cortex displaying stronger activity, and the same kind of profile for the firing rates during the delay period. Now, something interesting that we wanted to do is to explain or to try to explore the dependency of this phenomenon with 2 global parameters of the model. One of them is the global coupling strength, so overall, how strong is the interareal coupling between areas? And second, this level of feedback inhibition that I was telling you a few slides ago. So as you can see, if we want to have some level of distributed working memory, we have to avoid this corner. So meaning that if we have a lot of feedback inhibition and very little global coupling, we are not going to see these effects. And if we go to this corner with very few feedback inhibition and very strong global coupling, then we will have a lot of patterns. The best fit with the data, we found it more or less around here. But what is important, what surprised us, is that there is a little bit of something going on here. We call this a forbidden gap. And this is a region in the parameter space in which it's impossible to have distributed working memory. And this is, of course, something that surprised us a lot. And we started playing with the model. We realized that it was not a fluke. This was something real with a more systematic study. This is a very clear area in which we don't have persistent activity in the model. And we found out that this is mainly because of the influence of the heterogeneity in the local areas that we have in the modeling. In particular, the heterogeneity that we add by considering real data about NMDA receptor densities in each one of the areas. If we linearize or we remove this heterogeneity about NMDA receptors, this gap that we have here disappears entirely. So this tells us something about the importance of considering region-specific data to constrain our models, because many of the predictions that we can get out of this depend a lot on using high-quality data and specific data for each one of the regions. I'm going to finish with a couple of slides. I don't know if I'm 2 minutes, 3 minutes. Perfect. Last thing that I wanted to say is that something interesting that we can do with this kind of models, of course, these are the first batch of models in which we can look at cognitive functions like working memory in action in a large-scale model, not only at the level of the dynamics, but also at the level of the mechanism and the sustenance of selective information and all that. So now we can solve these kind of questions that have been haunting the field for several years. For example, one thing that we were interested in was to see, to try to say something about the polemic about whether primary visual cortex is able to sustain activity or to sustain, information regarding to working memory or not. Because indeed, when you are just looking at the voxel activity of primary visual cortex in a working memory task, you can actually decode the contents of working memory just by looking at primary visual cortex. You don't need to go to prefrontal cortex. So does it mean that V1 is actually sustaining information there, or is just reflecting what's happening in other places? Because now we have a working memory model that has multiple areas, we can test this. And the model is basically suggesting that, well, in terms of firing rate, V1 is going to be on this side. So there's not going to be persistent activity, just like we observe in the data. And we can very easily get the firing rates of the traces firing rate of V1, convert it into a Ball signal with the Balloon-Wien-Kessel model, and build a classifier that is able to tell us that, yes, indeed, just looking at the information that we have and the firing rates and the Ball signals that we have from V1 in the model, we are able to predict the contents of working memory, that is, whether we are remembering item A or B, with a very high level of accuracy, about 83%, which is in agreement with the levels that Harrison and Tong found in their experimental paper. And this is surprising because we know that V1 is basically receiving a lot of feedback inhibition from higher areas, but it also receives a little bit of excitation that is targeted to the particular populations. And one of them, the one that is sustained in higher areas, is going to be slightly stronger. One could think that this level is just too little to do something, but the model predicts that actually with just this level of differential excitation, you can actually discern the contents of working memory just by looking at visual cortex. So this supports the idea that visual cortex is somehow reflecting the information that is being stored more properly in higher areas, although, as I'm proposing now, not only in prefrontal cortex. Just the final slide, of course, is interesting to do a comparison. This is a workshop about comparison between species, right? So I just wanted to say that there are key differences between the distributed working memory patterns that we find in macaques and in humans. In general, the patterns in humans have comparatively less firing rates across all of the areas that we think are a consequence of a difference of the differences in the hierarchies that we use for the macaque and the human. And of course, like I showed you before, this kind of level of feedback inhibition is very important for the macaque, but the humans are way more robust for this, meaning that the feedback in humans doesn't have to be as constrained as for the macaques. We can have, of course, feedback inhibition, but we can have something a little bit more mild, let's say, and we can still get, for example, this clear separation between top hierarchy and bottom hierarchy without a lot of feedback inhibition. Okay, so just to conclude, just to get an idea of the importance of trying to look at cognitive functions at a more large-scale level. In this case, we propose the idea of a distributed working memory that works in several species. Feedback is more restrictive in macaques than in humans. And in the case of humans, we find these forbidden gaps in the parameter space, and we are able to explain the decoding of working memory contents from V1. If you want to have a takeaway from here, working memory seems to be a large-scale process rather than a localized process. It's not everything everywhere all at once, but it's almost that, because we know that some areas don't participate in that. And with that, just I wanted to thank everybody who helped with this. Xiaoying, of course, who was my mentor when I started working on this, and we got the first paper out. It's been in bioRxiv for a few years, but because of the magic of the pandemic, among other things, it's taking a while, but now it's out since last year. And the human study is something that is now on bioRxiv, thanks to my two students and postdocs that worked really hard on this. So I want to thank all of them, and also the students in my group, for all of the bombardment of ideas and support. Thanks.